Myosin is a protein with two globular heads pointing outwards and a long tail which contributes to the structure of the filament. The heads are the site of all the activity, since there are ATP binding sites, as well as actin binding sites, meaning this is where the thick and thin filaments will interact by making cross bridges. It's important to note the lack of myosin heads in the center of the sarcomere, just as there also are no thin filaments. Speaking of those, as we said, they are made predominantly of actin, and two actin filaments will twist together to form the backbone of the thin filament. Each actin subunit has an active site where myosin can bind, and when a muscle fiber is relaxed, these are blocked by spiraling strands of tropomyosin. There is also troponin, a globular complex of 3 polypeptides, one of which binds to actin, one of which binds to tropomyosin, and one of which binds calcium. Beyond this, we must be aware of the sarcoplasmic reticulum, a series of tubules that surround each myofibril. These regulate levels of calcium, which is needed for muscle contraction, through storage and release. This structure includes the T-tubules, which sit at each A-band, I-band junction, encircling each sarcomere and helping signals reach every region of the muscle cell. Now that we've examined each structural component, let's describe how everything works using the sliding filament model of contraction. This says that when the nervous system stimulates muscle fibers, the myosin heads on the thick filaments will interact with the binding sites on the actin subunits. These attachments will form and break several times as the thick filaments pull the thin filaments in towards the center of the sarcomere, thus pulling the Z-discs towards the M line. The I bands shorten and the H zone disappears. The overall effect is that the A bands from adjacent sarcomeres get closer together, so the entire muscle cell will shorten. This is the mechanism by which muscles contract. Now the question is, how is all of this activity initiated? As we said, this begins with a signal from the nervous system. We will examine that system in detail a bit later, but for now we can just examine the interface between the nervous system and a skeletal muscle. This is called the neuromuscular junction. Each muscle fiber has one, and these sections, called axon terminals, are nearly touching the muscle fiber, separated only by a thin space called the synaptic cleft, where the muscle produces